Hello guys, welcome back to our channel. We are on DUPariksha. Guys, I hope आपको हमारी जो कार्बोहाइड्रेट मेटाबॉलिज्म की हमने पूरी सीरीज बनाई थी यूनिट 1, यूनिट 2, यूनिट 3 एंड यूनिट 4 के जो PYQ सॉल्व करे थे वो आपके लिए हेल्पफुल रहे होंगे, आपको समझ आया होगा और आपको अच्छे से वो अच्छे लगते हैं। हमारे सीरीज अच्छी लगती हैं ये PYQs की सेमेस्टर 2 के जो हम सॉल्व कर रहे हैं BSc ऑनर्स बायोकेमिस्ट्री के। तो अपने दोस्तों के साथ जरूर शेयर कीजिए, हमारे चैनल को सब्सक्राइब करिए एंड लाइक करिए हमारी वीडियोस को। एंड टुडे वी विल बी सॉल्विंग NEP पेपर of cell biology of 2024. Cell biology, which is also a paper of the BSc Honors Biochemistry of semester 2 in DU. We will be solving this question paper, last year question paper of DU. Now let's see the first question. Name the following. An actin binding protein. Actin binding proteins regulate assembly and disassembly as well as organization of the actin filaments. इधर आपको नेम करना है कोई भी एक्टिन बाइंडिंग प्रोटीन। एक तो होता है ट्रोपोमाइसिन, फॉर्मिन, ARP 2, 3, आप इसमें से कोई से भी वन मेंशन कर दो। देन सेकंड वी हैव a protein forming nuclear lamina. The nuclear lamina is formed by what? Laminin proteins, which are a class of the intermediate filament protein. They provide what? Mechanical support as well as organized chromatin. Third is lysosomal storage disease, that is Gaucher's disease, that occurs due to deficiency of the enzyme, that is glucocerebrosidase. Fourth is the place of rhizosome assembly, that is nucleolus. And the last one we have the disease that is caused due to defect in peroxisome biogenesis, that is Zellweger syndrome, which is a rare disorder that is characterized by the reduction or absence of the functional peroxisomes in the cells of an individual. Let's look at the second part of the first question. We have to define the following terms. Here, first one is magnification. Magnification refers to the ratio of the size of final image that is obtained to the size of an object. It depends on the numerical aperture of the lenses as well as wavelength of the lens. Second is important. Important are the nuclear transport receptors that recognizes what? Nuclear localization signals, also known as NLS, that carry proteins into the nucleus by nuclear pore complex. It belongs to the family of the proteins that is involved in the transport of the molecules or proteins into the nucleus, known as caryophorins. Third is fixative. Fixatives are the chemicals that are used in microscopy, especially in preparing samples for light and electron microscopy. It helps to preserves and maintain integrity of the specimen, whether it's cells or tissues. It maintains structure or architecture of the molecular components of your sample. Works by stopping biological activity, stabilizing cellular components, preventing autolysis as well as decomposition. Fourth one is MTOC. MTOC stands for microtubule organizing center. It is a cellular structure from which microtubules are nucleated, anchored and organized. Centrosome is the primary microtubule organizing center in animal cell. ये ऑर्गेनाइज करते हैं माइक्रोट्यूबुलर साइटोस्केलेटन को बाय कंट्रोलिंग देयर नंबर, लोकेशन, ओरिएंटेशन एंड स्टेबिलिटी ऑफ द माइक्रोट्यूबुल्स. मोस्टली जो माइक्रोट्यूबुल्स होते हैं ना, दे आर एंकर्ड एट द माइनस एंड्स इन द MTOC और ग्रो आउटवर्ड्स towards the cell periphery, and centrosome as MTOC is located near the nucleus, where it is located near the nucleus, and it consists of a pair of centrioles that is surrounded by a pericentriolar matrix. Cell adhesion molecules, they are responsible for the interaction between cell and its environment or with other cells. They are present on the cell surface. So they are mostly cell surface protein. All adhesion molecules are the cell surface protein. Example, it is integrin. Now see the third part of the first question. First question, which is the compulsory question in the DU question papers. Here we have to give the location and the function of the following. First we have integrin. We have seen in the last question that integrin is a cell adhesion molecule that is involved in the cell interaction between its environment or with other cells. It is present on the plasma membrane of all cells except RBCs, and again it is involved in cell adhesion, cell locomotion as well as cell migration. Second one is catalase. Catalase is present in peroxisomes. It converts harmful hydrogen peroxide that is generated in our body due to metabolic processes is converted to the water and oxygen. It protects our cells from the oxidative damage. Now third one is connexin. Connexin is also present on the plasma membrane of the adjacent cell. It forms gap junctions. In this, connexin assemble to form connexons. These connexons dock with connexons of neighboring cells to form gap junctions. These channels allow direct exchange of the ions, small molecules and signaling molecules between the cells. Fourth is succinate dehydrogenase that is present in the mitochondrial inner membrane. Function, it is involved in ETC, electron transport chain, that transfers electron in the electron transport chain, that generate proton gradient for the production of the energy ATP, and it is also involved in TCA cycle. Now last one is keratin. Keratin is a structural protein. It is present in the cytoplasm of the epithelial cells. It contributes to structure like hair, nails and skin. It provides the strength to the cell. Let's move on to the second question. Here we have to differentiate between the following. First one is mitosis and meiosis. ये हम शुरू से ही करते हुए आ रहे हैं, like from 10th class. Mitosis single division होता है and it results in two daughter cells, two diploid daughter cells. Okay, while meiosis division involves two successive divisions, meiosis 1 and meiosis 2, and in this 4 haploid daughter cells are produced. Mitosis occurs in somatic cells while meiosis occurs in germ cells. Mitosis is known as equational division because the daughter cells have the same diploid number of chromosomes as the parent, because in this sister chromatids separate, while in meiosis, as we know, meiosis involves two divisions, meiosis 1 and meiosis 2. Meiosis 1 is known as reductional division because in this chromosome number is reduced to half, while in meiosis 2 sister chromatids separate and chromosome number remains the same, and it is known as equational division. What happens in mitosis? In this, prophase is short and it is not subdivided. It do not have substages, but in meiosis, prophase 1 is very long and it contains 5 stages: liptotene, zygotene, packetine, diplotene and dikinases. In mitosis, there is no crossing over, no pairing of the chromosomes and no chiasmata formation. So here in this, no genetic variation is seen, while in meiosis, in the zygotene stage of the prophase, homologous chromosomes pair and crossing over takes place during packaging, and chiasmata is formed in the diplotin stage. In mitosis, synaptonemal complex is not formed, while in meiosis, synaptonemal complex is formed during zygotine stage of prophase 1, and in meiosis, genetic variation is seen. What happens during the metaphase? In metaphase, plate alignment. In mitosis, chromosomes align singly, while in meiosis 1, homologous pair align. And during the anaphase, in mitosis, chromatids of each chromosome separate. During anaphase 1, homologous chromosomes separate. Now let's see second part of the second question, where we have to differentiate between the animal cell and the plant cell. Plant cells are usually longer than the animal cells, and in plant cell we have cell wall present in addition to the plasma membrane, and it also consists of the middle lamella, primary cell wall as well as secondary cell wall, while in animal cell wall is absent, and here we have only plasma membrane. In plant cell we have plasmodesmata, chloroplast, while in animal cell we don't have plasmodesmata and chloroplast. In animal cell, vacuoles are small or temporary, while in plant cell, what happens? Vacuoles are large and they are permanent for the storage as well as turgor pressure. Tonoplast, which is a membrane that surrounds vacuole, is present in the plant cell, while in animal cell it is absent. Centrioles are absent plant, but it is present in the motile cells of the lower plant, while in plant animal cells centrioles are present and they are involved in cell division. Nucleus is present along the periphery of the cell in the plant cell, while in animal cell nucleus is present at the center of the cell. Lysosomes are present in the animal cells. Lysosomes are rare in the plant cell. Storage material is usually starch granules in plant cells, while storage material is a glycogen granules in the animal cells. Write difference between peroxisomes and lysosomes. Peroxisomes are small membrane-bound organelles, while lysosomes are also single membrane-bound organelles. Peroxisomes contain more than 50 enzymes that are involved in various biochemical metabolic processes. Usually it contains oxidative enzymes, for example catalase, and lysosome contains degradative enzymes and acid hydrolysis. What is the function of the peroxisome? Peroxisomes are involved in lipid metabolisms and detoxification. They generate and break H2O2, that is hydrogen peroxide, by the activity of catalase. While function of the lysosomes are involved in the degradation of the macromolecules like proteins, lipids, and what they do? They operate at acidic pH and that is maintained by proton pumps. Peroxisomes is also involved by the lipids, and in liver it synthesizes bile acids, while lysosomes are involved in autophagy and they digest materials that are taken up by the endocytosis. Disease of peroxisome is Zellweger syndrome, and lysosomal storage disease is Gaucher disease. Endocytosis is a process of internalizing or taking up the material or substance into the cell, while exocytosis means process of expelling substances or materials from the cells. What happens in endocytosis? It involves invagination of plasma membrane to form vesicles surrounding the foreign material, while in endocytosis, in this fusion of the vesicles having waste with the plasma membrane occurs. Endocytosis involves import of nutrients or receptors. Endocytosis involves export of hormones, enzymes, and waste. In endocytosis, endocytic vesicles are formed like phagosomes. In exocytosis, secretory vesicles are formed. Endocytosis are of three types: phagocytosis, pinocytosis, and receptor-mediated cytosis. Receptor-mediated cytosis, through which receptor is taken up by the cells. And if we look at the types of the exocytosis, they are two: constitutive and regulated exocytosis. Example of the exocytosis is releasing of hormones out of the cell, and example of the endocytosis can be engulfing bacteria by phagocytes or macrophages. Gap junctions are present in animal cells while plasmodesmata is present in plant cells. Gap junctions are specialized intracellular connections between the adjacent animal cells. On the other hand, plasmodesmata are the cytoplasmic channels that connect plant cells through their cell walls. Gap junctions are composed of protein units called connexins. They assemble in hexameric form to form connexons. Connexons then dock with connexons of the adjacent cell, forming gap junctions. And plasmodesmata is made up of plasma membrane, lined channel, and desmotubules. Gap junction has no connection with endoplasmic reticulum, while plasmodesmata in this desmotubule, a membrane tube that is derived from the ER, connects the two endoplasmic reticulum. Plasmodesmata is involved in the transfer of water, ions, small molecules and large molecules also, like RNAs and some proteins, while gap junctions are involved in transfer of ions, small molecules and electrical signals. The function includes rapid communication and coordination between cells. If Let's see the function of the plasmodesmata, that facilitate communication and transport of materials. Last is a semi-autonomous organelle that has prokaryotic origin. As we know, chloroplast is a double membrane bound organelle that has a third additional membrane system, thylakoid membrane system, which forms a network of the flattened disc known as thylakoids. Then these thylakoids are arranged in a stack to form grana, and chloroplast is semi-autonomous organelle because it contains its own circular DNA, which is present in multiple copies per organelle, and chloroplast gene encodes for RNAs and proteins that functions in photosynthesis, and it also replicates independently within the cell by binary fission. But some of the proteins that are involved in the chloroplast are also encoded by nuclear genome, and why it is said to have prokaryotic origin. It is supported by endosymbiotic theory. According to endosymbiotic theory, chloroplasts are believed to have evolved from photosynthetic bacteria, cyanobacteria, that were engulfed by eukaryotic cells and formed a symbiotic relationship and evolved into chloroplast. They became a chloroplast. Why? Because why they are said to have prokaryotic origin? Because chloroplasts share many similarities. Cyanobacteria, which is a prokaryote, because it has circular DNA like bacteria, and it has 70S ribosome similar to bacterial ribosomes. It has double membrane, okay, and it divides independently like bacteria by binary fission. Chloroplasts also have a photosynthetic machinery that is similar to the prokaryotic photosystem, including thylakoids and pigments. Now let's see the third question, second part. Give the structure and function of the nuclear pore complex. As we know, the outer and the inner membranes are joined at the nuclear pore complex. It is the only channel for the transport of the different molecules between nucleus and the cytoplasm. It has a diameter of around 120 nanometer and it is composed of 30 different proteins, multiple copies of 30 different proteins. These are known as nucleoporins, and the nuclear pore complex has a 8 scaffold symmetry around the central channel. This is the central channel. In this, 8 spokes are arranged around the central channel, 4 here, 4 here. In this, 8 spokes extend from the nuclear channel that are connected by the nuclear rings on either side. एक nuclear ring इधर होती है cytoplasm side, एक nuclear ring इधर. Okay. These 8 spokes project radially from the rings. Structural components of the nuclear pore complex. First, we have cytoplasmic filaments. Cytoplasmic filaments, these are the cytoplasmic filaments, thread-like structure. They extend from the cytoplasmic ring and help in the cargo recognition and to capture the incoming cargo protein. These are anchored to the cytoplasmic ring. This is the cytoplasmic ring. Then we have a central channel in which nucleoporins are lying. They have multiple FG repeats, and at the base of the NPC we have nuclear ring, and there we also have a nuclear basket, that is the filamentous structure on the nuclear side that is involved in RNA export and assembly of the export complexes. In this, spoke ring assembly is anchored in nuclear envelope at the fusion of the inner membrane and outer membrane. प्रोटीन फिलामेंट्स हमने देख लिया कि साइटोप्लाज्मिक फिलामेंट्स एंड दी न्यूक्लियर फिलामेंट्स, ये दोनों एक्सटेंड करते हैं फ्रॉम बोथ दी साइड, जहां हमारी न्यूक्लियर साइड पे होती है बास्केट लाइक अपीयरेंस. पैसिव डिफ्यूजन ऑफ द स्मॉल मॉलिक्यूल्स that are less than 40 kilodalton. These can move freely across the pore in either direction. And the second is selective transport of the macromolecules. Like some proteins and some molecules require signal mediated transport and active transport of the macromolecules. It includes import and export. Import from the cytoplasm into nucleus and export from the nucleus into the cytoplasm. Nuclear localization signals on the cargo proteins are recognized by the importans. Importans are the nuclear receptors. They mediate the import of the what? Proteins or molecules into the nucleus by nuclear pore complex, and for the export, nuclear export signals on the export cargo protein are recognized by the importans, that are again nuclear receptors, and it is also involved in both of the mRNA. Now third part, what are actin binding proteins? How do they regulate the organization of the micro? Actin binding proteins are proteins that are involved and modulate the assembly and disassembly as well as organization of the actin filaments. Actin filaments also called microfilaments. Here is the table of the proteins that are involved in different cellular roles and the organization of the microfilaments. So there are various actin binding proteins like ARP23 and formin that bind along the length of the actin filament, that is responsible for the filament initiation and polymerization. Then nebulin and tropomyosin, filament stabilization, filament cross-linking. मतलब cross-linking one filament to another is done by alpha-actin, filament, vilin and fibrin. And capping is done by CAPZ and tropomodulin. They stabilize the microfilaments or actin filaments by capping the ends and preventing dissociation. Others promote dissociation, and they also regulate the exchange of ATP for ADP. For example, dissociation, ADF, coupling, thymosin, and propylene for monomer binding, as well as other protein also regulate the exchange of ATP for ADP. Now let's see the first is initiation of actin filament polymerization. ये कौन काम करते हैं? ARP and formulin. ये determine करते हैं where filaments are formed by facilitating nucleation, and nucleation is the rate limiting step in the actin filament polymerization. तो formin nucleate करता है long unbranched actin filaments को. It binds what? Actin, ATP actin, and nucleate initial polymerization of the actin monomers. Then formins move along the growing filament chain. ये देखो ये फॉर्मिन है, इससे हमारा एक्टिन मोनोमर बाइंड होते जाएंगे एंड प्लस ये प्लस एंड होता है ना, उससे उधर ऐड करते जाएगा न्यू मोनोमर्स और ये न्यूक्लिएट कर देगा अनब्रांच्ड एक्टिन फिलामेंट जो स्ट्रेस फाइबर्स बनाते हैं, कॉन्ट्रैक्टाइल रिंग बनाते हैं एंड थिन फिलामेंट्स ऑफ द मसल सेल्स. It is also associated with other protein, that is propylene, that is responsible for the exchange of bound ADP actin to the ATP actin. ARP23 complex here bind कर जाएगा किससे? बाइंड कर जाएगा, ये होता है हमारा ATP actin, इससे बाइंड कर जाएगा नियर द बार्ब्ड एंड, दैट इज प्लस एंड, और इनिशिएट कर देगा फॉर्मेशन ऑफ व्हाट? It initiates the formation of the branches because actin filament actively turn over and branch extensively. Then for the depolymerization, we have ATF covalence that modifies the existing filaments. ये हमारा filament है, microfilament. It enhances what? Rate of dissociation of the ADP actin. ये होता है ADP actin monomers from the pointed end, and they remain bound to the monomers that prevent their reincorporation. ये एक तरीके से कट कर देते हैं फिलामेंट्स को इनटू शॉर्टर पीसेस और प्रमोट करते हैं depolymerization from the pointed end. Then capping is done by cap C and tropomodulin. Two proteins, what they do, they bind to the filament के end पे bind करते हैं and prevent further polymerization and depolymerization. Then cross linking of one filament to another is done by fibrin and alpha actinin. They link filaments together into a network of bundles to provide structural support. This is how actin binding protein regulate the organization of the microfilament. Fourth part, what part of glucosaminoglycans allow them to form hydrated gel? Glucosaminoglycans are long linear polysaccharides having high density of negative charges, primarily from sulfate and carboxyl groups. They have a large number of negatively charged groups, so they attract a large number of cations, which in turn draw what? Water molecules by osmosis, and water bound to glucosaminoglycans create a gel-like structure, or you can say consistency. So they form gel-like consistency because of this, and allowing extracellular matrix to resist compression. Now fourth question, first part of the fourth question, define resolution and what factors affect the resolution of a microscope. Describe the different types of stain used in light microscopy. Resolution is the ability of a microscope to distinguish between two adjacent points. It is the degree of the clarity and details visualized The factors that affect the resolution of a microscope include numerical aperture. Numerical aperture is a measure of a lens ability to gather light and resolve fine specimen detail. जितना ज़्यादा numerical aperture होगा उतनी ही better resolution होगी. Then wavelength of light. Shorter wavelength means better resolution. Then we have refractive index of the medium. Refractive index. Immersion oils have higher refractive index than air. That reduces what? Light scattering or increase कर देता है resolution. Now describe the different types of stain used in light microscopy depending upon the different components of the cell, negative, basic. There are different. First one is basic stain. Basic stains are cationic. They carry positive charge. They are positively charged dyes, which means they will bind to negatively charged components of the cell, like nucleus that has RNA and DNA. Example: methylene blue and safranin and crystal violet. Then we have acidic stains. They are negatively charged dyes, anionic. They will bind to positively charged cell components in the cytoplasm. Example: eosine. Then we have neutral stains. They carry negative charge as well as positive charge also. They can stain both acidic and basic cell components. Example: Gimsas stain. Then fourth one we have vital stains. They are used for staining live cells. For example, genus green B for visualizing mitochondria. What are the different components of the extracellular matrix and explain briefly. So extracellular matrix fills the spaces between the cell and binds cell together. It provides the structural support, and the components of the extracellular matrix include structural proteins, collagen and elastin. These are the fibrous proteins. Matrix adhesion molecules. These are laminins and fibronectins that mediate cell attachment and signaling between the cells. Then we have matrix polysaccharides, glucosaminoglycans and proteoglycans. Now let's first see the structural proteins. That is, first is collagen. That is the most abundant protein in the ECM. It is composed of 3 polypeptide chains that are twisted into the triple helix like rope. Each polypeptide chain has repetitive glycine XY triplets where proline and is X and Y is usually hydroxyproline. Because of their ring structure, ये amino acid stabilize करते हैं helical conformation of the polypeptide chain. Collagen provide tensile strength and structure to the tissues like skin, bones, and tendons. Collagens are a large family of protein containing more than 40 different protein. Type 1 in collagen is most abundant. Type 1, type 2, type 3 and type 5 are fibrillar collagens. These are connective tissue proteins that provide tensile strength, and they are usually found in bones, skin, tendons, cartilage and arteries. Then collagen 4 forms network or sheets. They provide flexibility to the collagen network and to the basement membrane. Then the second structural protein in ECM is elastin. Elastin is usually found in the tissues that requires elasticity and that needs to stretch and recoil, such as bladder, lungs, ligaments. They are composed of soluble tropoelastin protein with containing glycine, valine, and modified aline and proline residues. They are highly cross-linked into a network by covalent bonding between the side chain of lysine residue, because of which they behave like a rubber band that can be stretched under the tension and then snapping back when tension is released. Then we have matrix polysaccharides. Now for matrix polysaccharides, we have glucosaminoglycans. So fibril structural protein of the ECM are embedded in the fibril structural proteins of the ECM are embedded in the cells that are formed from polysaccharides called glucosaminoglycans. They are long polysaccharides which consist of repeating units of disaccharide. One sugar of the disaccharide is either N-acetyl glucosamine or N-acetyl galactosamine. It also has acidic sugars, glucuronic acid or ioduronic acid. These are modified by the addition of the sulfate group with an exception of the hyaluronan, which is not modified by the addition of the sulfate group. GAGs form hydrated gels because of the high density of the negative charges, either carboxyl group or sulfate groups. So they attract what cations from the environment and they trap water molecules by osmosis. So they form hydrated gels, thereby providing mechanical support to the extracellular matrix. The most common GAGs are chondroitin sulfate, dermatan sulfate, keratin sulfate, and heparin sulfate. Okay, heparin sulfate. So we also have proteoglycans. Proteoglycans have a protein core to which glucosaminoglycans are attached, and they provide water resistant to compression and anchors cell to the matrix file. Then we have adhesion proteins, the third class of the ECM constituents. They are responsible for linking components of the matrix to one another and to the surface of the cell. They also interact with collagen proteoglycans to specify what matrix organization और एक तरीके से मेजर बाइंडिंग साइट देते हैं फॉर सेल सरफेस रिसेप्टर्स सच एज इंटीग्रिन्स. तो पहला आता है हमारा fibronectin, which is a glycoprotein dimer. It means it has two polysaccharides and it is linked to fibrils. It has binding sites for collagen and proteoglycans. That's why it forms cross-links with matrix components. It also has binding sites for integrins. Then we have laminin. Laminin family. ये एक तरीके से कहाँ मिलते हैं? बेसल लैमिना में मिलते हैं. दे आर इधर क्रॉस शेप्ड और टी शेप्ड हेटेरोट्राइमर्स होते हैं जिसके अंदर अल्फा सब यूनिट भी होता है, बीटा सब यूनिट भी होता है एंड गामा सब यूनिट. एंड दे हैव बाइंडिंग साइट फॉर इंटीग्रिन्स, प्रोटियोग्लाइकन्स as well as proteoglycans, heparin sulfate, and for domain binding site for collagen type 4. They also act as they are responsible for the cell adhesions, movement and anchoring glycoprotein to the basal laminae, and it is also involved in cell differentiation. Third part of the fourth question, explain the process of assembly, organization, and the role of intermediate filaments. Intermediate filaments are the cytoskeletal filaments of about 10 to 12 nanometers. So, एक जो मोनोमर होता है सिंगल पॉलीपेप्टाइड में देखा जाए तो इंटरमीडिएट फिलामेंट के पास सेंट्रल अल्फा हेलिकल रॉड डोमेन होता है एंड इट आल्सो हैज वेरिएबल टेल एंड रीजन, हेड एंड टेल रीजन. ये जो अल्फा हेलिकल रॉड डोमेन होता है इसके अंदर अप्रॉक्स 310 अमाइनो एसिड्स होते हैं. तो अब इसकी असेंबली देखते हैं कैसे बनती है. So from the monomer, two monomers form dimers. They coiled around each other in a parallel arrangement or they form a dimer. So after formation of the dimer, two dimers associate in anti-parallel or staggered manner to form a tetramer. No polarity because both ends are equivalent, so they are apolar. After that, profilaments are formed. Tetramers then assemble end to end to form profilaments. After that, 8 profilaments wind around together in a rope-like structure, or they associate laterally in a rope-like filament that is 10 nanometer thick to form final filament. Each protofilaments bind together in a rope-like structure to form what? Final intermediate filament. Now their organization in the cell. So intermediate filament forms a network that anchors and support internal structure extending from nucleus to the plasma membrane. It is also attached to other cytoskeleton elements such as it bridges with actin filaments and microtubules by lectin, and it is also associated with cell junctions like desmosomes and hemidesmosomes. Functions include it provides mechanical strength to the epithelial tissues and resists stretching, compression, and helps to maintain the cell shape, resist deformation again, and it is also involved in the support of the axon. Neurofilaments support long axons in neurons as well as Desmin links contractile units in muscle cells. Now let's see the fifth question. First part, A part, give principle and merits of the phase contrast microscopy over bright field microscopy. So the principle of the phase contrast microscope is based on the interference between undiffracted light and the diffracted light. Undiffracted light is the light that passes through the specimen without change, and diffracted light is the light that slows down or shifts space slightly due to passing through structures in the specimen. In this, we separate the direct light that enters the specimen objective lens from the diffracted light emanating from the specimen. By this, we get an image with highly contrast bright and dark areas against well illuminated background. Happens in phase contrast microscopy, we get a high contrasting image even in unstained specimen, but in bright field microscopy we get poor contrast for unstained specimens. We can examine live cells like mitochondria in phase contrast microscopy, but for bright field microscopy it is not. We can't view live cells, and we know phase contrast microscopy uses phase interference to enhance image, while ब्राइट फील्ड माइक्रोस्कोपी की जो इमेज होती है ना, इमेज की जो रेजोल्यूशन और जो उसकी कंट्रास्ट रे सब किस पे डिपेंड करता है, लाइट पे और लाइट के रिफ्लेक्शन पे. फेस कंट्रास्ट माइक्रोस्कोपी फाइन इंटरनल स्ट्रक्चर्स की डिटेल्स देख सकते हो आप इसमें. वाइल इन दिस अगर हम ब्राइट फील्ड माइक्रोस्कोपी में देखें तो लिमिटेड डिटेल्स से ही मिलेंगी हमें ट्रांसपेरेंट सेल्स, मतलब इसमें हमें स्टेन करना पड़ता है, लेकिन फेस कंट्रास्ट माइक्रोस्कोपी में ऐसा नहीं होता. B part explain the role of REN-GTP in the regulation of the nuclear import and export. So REN is a small GTP binding protein and it plays a central regulatory role and controls what directionality of the movement or transport across the nuclear pore complex. There is high concentration of the REN-GTP in nucleus, and REN-GTP is predominant in the cytoplasm. So asymmetrical distribution of REN-GTP and REN-GTP across the nuclear envelope is what drives and regulates both import and export of proteins through nuclear pore complex. Now let's see what happens in nuclear import. So in the cytoplasm we have a cargo protein with, it is a cargo protein which has nuclear localization signals that are recognized by what importance. These are the nuclear receptors. So they bind to the important, and a cargo important complex is formed and which moves through NPC. After Coming to nucleus, important and cargo protein complex separate. How? In the nucleus there is RAN-GTP that binds to important and causes a conformation change that will lead to the release of the cargo protein in the nucleus. So after the formation of important and RAN-GTP complex, this complex leaves the nucleus through nuclear pore complex, and in the cytoplasm this RAN-GTP is hydrolyzed to RAN-GDP, and which causes RAN to release important into the cytoplasm, which allow it to bind to new cargo protein. And so the RAN-GTP that is transported to the cytoplasm is taken back into the nucleus by its own receptor, important receptor, that is anti-F2. It will bind to RAN-GDP where and comes to nucleus. In the nucleus there is guanine nucleotide exchange factor that will generate RAN-GTP from RAN-GDP. This is how RAN-GTP is recycled back into the nucleus. So as we have discussed how nuclear import takes place, so let's revise the role of the RAN-GTP in the nuclear import complex. So RAN-GTP करता क्या है? It releases cargo protein from important by binding to important in what? In nucleus. It forms RAN-GTP important complex which is then transported back to the cytoplasm और release कर देता है important ताकि वो दूसरे कार्गो प्रोटीन से बाइंड करें और उन्हें ट्रांसपोर्ट करें into the nucleus. In the cytoplasm RAN-GTP हाइड्रोलाइज हो जाता है into RAN-GTP और वही important release हो जाता है for another round of import. Now we have nuclear export. Here's the diagram. So जो proteins that are meant to leave the nucleus contain nuclear export signals. ओके ये जो न्यूक्लियर एक्सपोर्ट सिग्नल्स होते हैं ना उनके पास ये रिच होते हैं इन ल्यूसीन और हाइड्रोफोबिक अमाइनो एसिड्स में. दे आर रिकॉग्नाइज्ड बाय व्हाट? इम्पोर्टेंट एक्सपोर्टेंट. सी दिस इज द कार्गो प्रोटीन विद न्यूक्लियर एक्सपोर्ट सिग्नल्स and it will be recognized by the exportin. This is exportin, and here we have RAN-GTP. RAN-GTP promotes what? Binding of the cargo protein and exportin into a stable trimeric complex. ये देखो ये trimeric complex फॉर्म होगा RAN-GTP, cargo protein and exportin. ये लीड कर जाएगा. This complex travels from the nucleus through the nuclear pore complex into the cytoplasm. वहां क्या होगा? इन द साइटोप्लाज्म वही RAN-GTP हाइड्रोलाइज हो जाएगा इनटू RAN-GTP, जिसकी वजह से हमारा कार्गो प्रोटीन रिलीज होगा एंड एक्सपोर्टिन रिलीज हो जाएगा. एक्सपोर्टिन और जो RAN-GTP होता है ना ये दोबारा रिसाइकल्ड हो जाते हैं. दे विल रिटर्न टू द न्यूक्लियस फॉर रीयूज. फिर वही RAN-GTP इज रिचार्ज्ड बाय और इज रीजनरेटेड इनटू RAN-GTP बाय व्हाट? Guanine nucleotide exchange factor in the nucleus. Revise the role of RAN-GTP in the nuclear export. First ये क्या प्रमोट कर रहा है? बाइंडिंग ऑफ द एक्सपोर्टिन टू द कार्गो प्रोटीन, और स्टेबलाइज करता है ये जो ट्राइमेरिक कॉम्प्लेक्स फॉर्म होता है ना हमारा कार्गो एंड एक्सपोर्टिन कॉम्प्लेक्स. इसके बाद फिर ये इसको रिलीज कर देता है इनटू द साइटोप्लाज्म. जब ये रिलीज कर देगा साइटोप्लाज्म में तो हाइड्रोलाइज हो जाता है RAN-GTP में और कार्गो प्रोटीन रिलीज एंड एक्सपोर्टिन दोबारा रिसाइकल बैक हो जाता है इनटू द न्यूक्लियस. Describe the organization of the tight junctions and why are tight junctions important for maintaining integrity of the polarized. Tight junctions are the specialized cell-cell junctions that are found near the apical surface of the epithelial cells. और ये एक तरीके से कॉन्टिन्यूस बेल्ट बनाते हैं जो सील कर देता है सेल को. या और दे ऑल्सो फंक्शंस एज अ बैरियर एज वेल एज ये होल्ड करेंगे सेल्स को टुगेदर और मेंटेन करते हैं पोलैरिटी ऑफ दी सेल्स बाय प्रिवेंटिंग व्हाट? लेटरल डिफ्यूजन ऑफ इंटीग्रल मेम्ब्रेन प्रोटीन बिटवीन apical and lateral basal surface. Tight junctions consist of web-like strands of transmembrane proteins. We can say that tight junctions are made up of network of sealing strands जो completely encircle कर देते हैं सेल के अराउंड, एंड दे आर composed of transmembrane protein. ये प्रोटीन इंटरैक्ट करते हैं विद सिमिलर प्रोटीन्स ऑन एडजेसेंट सेल, क्रिएटिंग अ टाइटली सील्ड बैरियर. तो मेजर जो हमारे ट्रांसमेम्ब्रेन प्रोटीन होते हैं उसमें आता है क्लॉडिन, ऑक्लूडिन एंड जैम, दैट इज जंक्शनल एडेंशन प्रोटीन. So what does occludin do? It regulates barrier functions और interact करता है with other tight junction components जो दूसरे सेल्स के टाइट जंक्शन कंपोनेंट्स होते हैं, और ये क्लॉडिन्स के साथ इंटरैक्ट करता है स्कैफोल्डिंग प्रोटीन्स के साथ. और वी हैव क्लॉडिन्स, वही ये परमेबिलिटी बैरियर की तरह काम करता है, एक तरीके से बैकबोन होता है टाइट जंक्शन स्ट्रेंथ्स का. while we have junctional adhesion proteins, they also help in cell adhesion. एंड ये जो साइटोप्लाज्मिक टेस्ट होती है ऑफ क्लॉडिन एंड ऑक्लूडिन, ये इंटरैक्ट एंड जैम, ये इंटरैक्ट करती है विद व्हाट, स्कैफोल्डिंग प्रोटीन्स के साथ. स्कैफोल्डिंग प्रोटीन सच एज ZO1, ZO2, ZO3. These are zonula occludens proteins. Zonula occludens of protein. ये जो ZO और ये जो जोना ऑक्लूडेंस प्रोटीन्स होते हैं ना, ये एंकर करते हैं व्हाट, टाइट जंक्शंस टू द एक्टिन साइटोस्केलेटन, और ऑर्गेनाइज करते हैं पूरा जंक्शनल कॉम्प्लेक्स को, और अडेप्टर्स की तरह काम करते हैं, लिंकिंग ट्रांसमेम्ब्रेन प्रोटीन्स टू वेरियस signaling pathways. They also forms a belt of transmembrane protein जो क्लोज कर देते हैं एक्स्ट्रासेलुलर स्पेस बिटवीन द सेल्स, जिसकी वजह से ये क्या करते हैं, प्रिवेंट करते हैं पैसेज ऑफ द वाटर एंड वाटर सॉल्युबल सब्सटेंसेस, एंड इट इज आल्सो रिस्पांसिबल फॉर व्हाट, इलेक्ट्रिकल रेजिस्टेंस इन epithelial cells, एंड आइसोलेट पार्ट्स ऑफ द वही पोलैरिटी मेंटेन करते हैं, लाइक एपिकल मेम्ब्रेन को, बेसिलेटरल डोमेन्स को सेपरेट कर. व्हाई आर टाइट जंक्शंस important for maintaining integrity of the polarized cell? As we know, tight junction isolate कर देते हैं parts of the plasma membrane into apical domain and basolateral domain, जिसकी वजह से ये prevent करते हैं lateral diffusion of the protein and lipids between apical and basolateral membranes, that allows specialized functions of each domain, that is apical and basolateral domains, to be conserved. Then it also acts as a barrier functions, why prevent the passage of the molecules and ions through the space between plasma membranes of the adjacent cells. By this, इसकी वजह से ये क्या करते हैं, prevent करते हैं leakage of the molecules between the compartments. Now the last question, that is sixth question. Write short notes on the following. First we have mitochondrial genome. So what is mitochondria? Mitochondria is a double membrane bound organelle जिसके पास एक आउटर मेम्ब्रेन होती है, इनर मेम्ब्रेन होती है. जो इनर मेम्ब्रेन होती है वो न्यूमरस फोल्ड्स बनाती है, क्रिस्टी, नोन एज क्रिस्टी, जो एक तरीके से एक्सटेंड होती हैं इनटू द माइटोकॉन्ड्रिया टुवर्ड्स द मैट्रिक्स. फिर इसके पास एक मैट्रिक्स होता है जिसका नाम एंजाइम्स, प्रोटीन्स, डीएनए. ये एंजाइम्स ऑक्सीडेटिव फॉस्फोराइलेशन, and it is also involved in TCA and various reactions. But mitochondria के पास खुद का genetic system होता है, and it has its own genome, that is circular DNA, which is present in multiple copies per organelle. Mitochondrial genome is mostly similar to the alpha proteobacteria, जो इनकोड करता है कितने प्रोटीन्स के लिए, 6700 to 8300. देन माइटोकॉन्ड्रिया का जो साइज होता है ना डीएनए का, वो वेरी करता है बिटवीन डिफरेंट स्पीशीज, इट डिपेंड्स अपॉन द डिफरेंट स्पीशीज. सो द लार्जेस्ट माइटोकॉन्ड्रियल जीनोम इज प्रेजेंट इन Arabidopsis thaliana, that is 367 kb. So most mitochondrial genome encodes for proteins that are essential components of the oxidative phosphorylation, and human mitochondrial genome encodes for 13 proteins that are involved in ETC and oxidative phosphorylation. Human mitochondria empty here is mitochondrial DNA. It encodes for 16S, 12S RNAs and 22 RNAs. Okay, 22 tRNAs. So the second one is functions of SER. Smooth endoplasmic reticulum, वही it is endoplasmic reticulum जिसके सरफेस पे राइबोसोम्स नहीं होते. Smooth endoplasmic reticulum is involved in lipid metabolism. इसके अंदर enzymes होते हैं to metabolize various lipid soluble components and various lipids are synthesized on the smooth endoplasmic membrane. फिर वो जब वो synthesize हो जाते हैं ना, they are transported to other membranes. So it is the main site for the synthesis of the phospholipids, cholesterol and steroid hormones. जैसे हमें इसके अंदर सिंथेसाइज होते हैं लिपिड सिंथेसाइज होते हैं, तो लिपिड्स हमें पता है हाइड्रोफोबिक है, वेदर दे आर फॉस्फोलिपिड्स एंड कोलेस्ट्रॉल. ठीक है, ये मेम्ब्रेन लिपिड्स का सिंथेसाइज हमारे स्मूथ एंडोप्लाज्मिक रेटिकुलम में होता है. तो दे आर हाइड्रोफोबिक, दैट्स व्हाई दे आर सिंथेसाइज्ड इन द SER membrane rather than in the cytosol. So most phospholipids that are derived from the glycerol are synthesized on the cytosolic side of the SER membrane. ये localization allow करता है क्या, हाइड्रोफोबिक फैटी एसिड चेन्स टू रिमेन बरीड इन द membrane. फिर आता है कि जब हमारा synthesize हो जाते हैं lipids, so they are transported to other membranes through what? Through direct membrane contact or by the formation of the vesicle, or third one by lipid protein transfers. Lipid transfer protein. So it also play a role in membrane growth. As lipid bilayer grow होती रहेगी, जैसे जैसे new phospholipids add होते रहेंगे. It also contains various enzyme, वही कि जो metabolize कर देता है various soluble compounds को, तो detoxification में भी कुछ मदद करता है. Then steroid hormones are synthesized. You guys can take a screenshot of these short notes. Functions of SER as well as mitochondrial genome. ये आप इसका स्क्रीनशॉट ले लो, वरना वैसे हम आप चाहो तो इसका स्क्रीनशॉट ले लो, लेकिन हम टेलीग्राम ऐप पर भी अपलोड कर देंगे ये नोट्स. इट्स लाइक इम्पोर्टेंट पॉइंट्स, ये सब इम्पोर्टेंट पॉइंट्स अगर आप ये याद रखोगे तो आप. नाउ फोकल अटेंशन. सो फोकल अटेंशन आर द स्पेशलाइज्ड स्ट्रक्चर्स. Now focal adhesions. So what are focal adhesions? Focal adhesions are the large dynamic protein complex जो कनेक्ट करते हैं सेल्स के साइटोस्केलेटन को टू द एक्स्ट्रासेलुलर मैट्रिक्स बाय इंटीग्रिन्स. Okay. So the structural components, if we see the structural components, we have integrins. You can see here, these are integrins. These sea blue and purplish blue color. Integrins are the transmembrane protein जो connect करते हैं सेल का interior to the ECM. Then we have talin and vinculin. This is talin protein and this is vinculin protein that connects what? ये link करता है यहाँ जो हमारा integrin है ना, उसको to the actin filaments. ये actin filaments है. Then we have alpha actinin that is responsible. This is alpha actinin. ये किस लिए रिस्पॉन्सिबल है? ये क्रॉस लिंक करता है बिटवीन एक्टिन फिलामेंट एंड और स्टेबलाइज कर देता है एक्टिन फिलामेंट्स को. देन वी हैव एक्टिन फिलामेंट्स. दिस इज एक्टिन फिलामेंट. ये साइटोस्केलेटल स्ट्रक्चर्स होते हैं जो हमने लास्ट इस पेपर जब हम NEP के क्वेश्चन सॉल्व कर रहे थे BSc पेपर में, तो हमने देखा था एक्टिन फिलामेंट्स इन्वॉल्व होते हैं सेल शेप में, मूवमेंट में. तो इसके अंदर हो क्या रहा है ये देखो. फोकल अटेंशन का अगर हम ये एक मैग्नीफाइड व्यू है नीचे, इसके अंदर क्या होता है, इंटीग्रिन्स हमारे ट्रांसमेम्ब्रेन प्रोटीन होते हैं जो स्पैन करते हैं पूरी मेम्ब्रेन को और बाइंड करते हैं टू द एक्स्ट्रासेलुलर मैट्रिक्स. दिस इज एक्स्ट्रासेलुलर मैट्रिक्स लाइक टू फाइब्रोनेक्टिन और लैमिनिन, इससे बाइंड कर जाएंगे. बट इनसाइड द सेल, ये हमारा साइटोप्लाज्मिक साइट है सेल का, इट इज साइटोप्लाज्म. ये जो टेलिन प्रोटीन होता है ना, ये कनेक्ट कर देता है इंटीग्रिन्स का साइटोप्लाज्मिक टेल हो गई ये कनेक्ट होगी टेलिन से, और टेलिन किससे कनेक्ट होगा? एक्टिन फिलामेंट से। तो ये एक तरीके से कनेक्ट कर देता है इंटीग्रिन्स टू द एक्टिन फिलामेंट। इट ऑल्सो हैड विनक्यूलिन एंड अल्फा एक्टिनिन जो रीइंफोर्स करते हैं इस कनेक्शन को। देन स्ट्रेस फाइबर्स दैट इज बंडल्स ऑफ द एक्टिन, ये स्ट्रेस फाइबर्स, ये बंडल्स ऑफ द एक्टिन, ये भी एंकर्ड होते हैं टू दिस कॉम्प्लेक्स। अगर हम इसके फंक्शंस देखें, यहाँ मैंने कुछ पॉइंट्स लिखे हैं, आपको फंक्शंस थोड़े एक्सप्लेन करने हैं। देखो, ये प्रोवाइड करता है मैकेनिकल स्ट्रेंथ, कैसे? ये फिजिकली एंकर करता है साइटोस्केलेटन लाइक एक्टिन स्ट्रेस फाइबर टू द ईसीएम, जिसकी वजह से मैकेनिकल सपोर्ट मिलता है। फिर ट्रांसडक्शन में काम करता है, कैसे? एक तरीके से सिग्नलिंग सेंटर की तरह सर्व करता है, हेल्प करता है सेल को टू रिस्पॉन्ड टू द एक्सटर्नल सिग्नल्स लाइक केमिकल स्ट्रेस और मैकेनिकल स्ट्रेस, या गया। एंड मोस्ट इम्पॉर्टेंटली सेल मूवमेंट, ये जो फोकल अटेंशन होते हैं ये फॉर्म करते हैं और डिसअसेंबल करते हैं डायनेमिकली ड्यूरिंग सेल माइग्रेशन। देन स्ट्रक्चरल इंटीग्रिटी, वही मेंटेन करते हैं स्ट्रक्चरल शेप, क्योंकि इसके अंदर एक्टिन फिलामेंट है, इट मींस सेल शेप को मेंटेन करेंगे, ओरिएंटेशन को by resisting contractile forces from the stress fibers. Now fourth one, treadmilling of the microfilaments. So what are, first we will see what is microfilaments. Microfilaments वही एक्टिन फिलामेंट्स, तो एक्टिन मोनोमर्स होते हैं जो जी एक्टिन्स होते हैं, ये पॉलीमराइज हो जाते हैं टू फॉर्म फिलामेंटस एक्टिन दैट इज एफ एक्टिन। तो फिलामेंट जो होता है ना, माइक्रोफिलामेंट्स और एक्टिन फिलामेंट्स, उनकी दे हैव पोलैरिटी, एक एंड इनका प्लस एंड होता है दैट इज बार्ब्ड एंड, एंड अदर वन इज द माइनस एंड दैट इज पॉइंटेड एंड। तो ट्रेडमिलिंग ना एक डायनामिक प्रॉपर्टी होती है, डायनामिक प्रोसेस होता है इन विच देयर इज कंटिन्यूस addition of the actin monomers at the barbed end that is plus end and loss of the monomers from the negative end. So at वही treadmilling is a dynamic process at cellular actin concentration. Barbed end जो होता है वो हमारा grow करता है 5 to 10 times faster than the pointed end because in this जो हमारा एटीपी एक्टिन होते हैं ना वो टाइटली बाउंड नहीं होते टू द पॉइंटेड एंड, सो दे डिसोसिएट एंड आफ्टर डिसोसिएशन दे then convert into ATP actin and this ATP bound G actin monomers, ये आ गया ATP bound actin monomers add हो जाता है to the pointed end known as barbed end. आफ्टर इनकॉर्पोरेशन एटीपी का हाइड्रोलिसिस हो जाता है, वो फिर कन्वर्ट हो जाता है एडीपी में। तो एक तरीके से बार्ब्ड एंड की तरफ से ग्रोथ होने लगती है और एटीपी एक्टिन की तरफ से डिसोसिएट होने लगते हैं मोनोमर्स। तो इसका जो रिजल्ट होता है ना वो ये आता है कि देयर इज नेट एडिशन इन द प्लस एंड एंड नेट लॉस इन द माइनस एंड, पर इनकी पूरी फिलामेंट की जो लेंथ होती है ना स्टेज द सेम। इसके अंदर जो एटीपी हाइड्रोलिसिस होती है ना ये एनर्जी के लिए नहीं होता, बट इट अफेक्ट्स द स्टेबिलिटी ऑफ द एक्टिन विद इन द फिलामेंट। और ट्रेडमिलिंग अलाउ करती है, ये जो ट्रेडमिलिंग इनकी प्रॉपर्टी होती है, इट अलाउज कंटिन्यूअस रिन्यूअल ऑफ द एक्टिन फिलामेंट while maintaining their length. And why treadmilling is important? एक तो सबसे पहली बात सेल मूवमेंट में, okay, and maintaining cell shape. Last one we have phagocytosis. So phagocytosis is a type of endocytosis in which material is taken up by the cells by the formation of the finger-like projections known as pseudopodia. In this cell extends finger-like projections, pseudopodia, around the material outside the cells. Then this pseudopodia eventually surround the material और एक तरीके से ज्वाइन कर जाएगी, और इसकी जो मेम्ब्रेन होती है ना pseudopodia की, that will join or fuse to form large intracellular vesicles in the cytoplasm known as phagosome. ये बन गया phagosome. फिर ये phagosomes के साथ फ्यूज हो जाएगा इन lysosomes. It will eventually form what? Phagolysosomes, जिसमें क्या होता है, in which material are ingested by what? by the action of lysosomal acid hydrolysis, in which जो हमारे large particles होते हैं वो smaller particles में convert हो जाते हैं. Materials that are ingested by the lysosomal acid hydrolysis are either used up by the cell और expelled हो जाते हैं as waste. इनका फंक्शन क्या है फैगोसाइटोसिस का? फर्स्ट अमीबा यूज करता है WBC डिफेंस मैकेनिज्म के लिए टू कैप्चर फूड और रिमूव करता है डेड सेल्स को. आज के लिए इतना ही. दैट्स इट फॉर टुडेज लेक्चर. आई होप आपने एंजॉय करा होगा, आपको मजा आया होगा और आपको समझ आया होगा. अगर आपको लगता है कि हमने कहीं मिस्टेक करी है, हमने किसी तरीके से हम अपना आप को इम्प्रूव कर सकें और आपको लगता है कि हमें कुछ चीज़ों को ऐसे करना चाहिए, तो आप प्लीज़ फीडबैक दीजिए. टेलीग्राम ऐप पर दीजिए या कमेंट सेक्शन में ज़रूर कमेंट करिए, क्योंकि आपकी राय ही हमारे लिए बहुत इम्पोर्टेंट है. आपके अकॉर्डिंग ही हम चीज़ों को कस्टमाइज करेंगे. अगर आपको हमारी वीडियोस अच्छी लगती हैं, आपको DU के PYQs यहाँ मिलते हैं, तो प्लीज़ अपने दोस्तों के साथ शेयर करिए, हमारे चैनल को सब्सक्राइब करिए एंड आपको मिलते हैं हम नेक्स्ट वीडियो में.